Hello friends, this video on locomotion and movement part 9 is brought to you by examfear.com. No more fear from exam. So we will now try to understand the structure of both the contractile proteins, actin and myosin. So we will start with actin. Let us try to understand the entire structure of actin, because only when we know the structure of actin and myosin we will be able to understand the concept of contraction. Okay, so actin is a polymerized protein. So what do we mean by polymerized protein? What do we mean by the process of polymerization? So polymerization is a process where lot of smaller monomers unite together or join together to form a bigger polymer. So that means actin is a polymer. That means it has several smaller units or several monomers which are combined together to form what we have, that is actin. Now, which are the monomers for this actin structure? Okay, actually actin is made up of monomer which are known as globular actins. So the monomers are globular in nature. Globular means spherical. So there are, which is also known as G-actins. So this is how they look like. So there are monomers like this, the globular actins. They are all single proteins. Now some of these join together to form bigger units. For example, two monomers join together to form a dimer. Similarly, 3 monomers join together to form a trimer. That is 3 monomers together, so it is a trimer. Similarly, several monomers join together to form a polymer. So this actin which we are talking about is a monomer unit which is formed in the shape of a helical structure. So several globular proteins are arranged in this helical fashion, and this is known as the actin, which we will be studying here. So composition of actin, it consists of helically wound 2 filamentous actin, which is known as F-actins. So this chain which is formed by the globular actins, so this polymer which is formed is known as the filamentous actin or F-actin. So you can say that F-actin is the polymer which is formed by the monomers G-actins. So F-actin is a polymer of G-actin in simple words. So the structure of the actin, it consists of 2 F-actins which are helically wound around each other. So here in this picture you can see this is one chain and this is another chain. So it is something like this. This is one chain and this is another chain. So that is how it is helically wound around each other. So this each circle represents a globular protein. So this is your F-actin. What else does it contain? So the monomers for this F-actins are the globular proteins or the G-actins. It also contains 2 filaments of a protein called tropomyosin. So here you can see there is another helical arrangement of another chain. This chain-like structure or this filament is another protein called tropomyosin. So tropomyosin, again 2 filaments are present in a similar fashion. And it contains another protein called troponin which is present at regular intervals. So this protein structure which you see here, that is troponin. So you can see that this protein is again present at regular intervals. So at specific intervals they are present. Now what is the purpose of this troponin protein? Now this protein masks the active binding site for myosin. Now this actin, as you saw even in the resting phase, there is some partial overlap between actin and myosin. That means actin and myosin, they combine with each other. Now how do 2 proteins can combine with each other? For that they need to have specific binding sites. It is exactly like those puzzles. You remember we spoke about those puzzles which fit into each other only at specific points. For example, this is your puzzle block. This is one puzzle block. This is another puzzle block. So these 2 puzzle blocks can fit in only at this site. So this is the active binding site for puzzle A and B, if this is A and this is B. So what is similarly in actin, there are specific active binding site like this where myosin can come and combine. But these sites have been blocked by these proteins, troponin. So as long as the troponin proteins are present, myosin cannot really combine or form a bond or cannot really connect good enough with actin. So troponin masks the active binding site of actin so that myosin cannot bind to actin. So this is the structure of actin. So actin, structure wise, it consists of three things. Two filamentous actins, that is these chains, these are the F actins. It consists of two filaments of tropomyosin, and it contains another protein, troponin, which masks the actin binding sites. So as I said, troponin masks the active binding site for myosin on actin filaments during the resting phase. So during the resting phase is that phase where a complete overlap does not take place, and that complete overlap does not take place because of the presence of troponin. So this is your puzzle blocks. This is one puzzle block, this is another puzzle block, right? Now let us say that this is the green one is actin. Let us suppose this is the actin filament and it has this site. This is the active binding site for myosin on actin. So this is the active binding site. Just understand this concept. Okay, and let us suppose this red one is the myosin. Now what happens if this red one comes? It can actually bind to actin at this active binding site. That is the meaning of active binding site. But now, if instead of myosin coming and binding to actin, if instead of that, there is something else which is coming and binding to this site. Now, what is this something else? This something else is troponin. So this troponin is actually masking the active binding site, and as a result the myosin now cannot connect with it because the troponin does not have a suitable site for myosin to bind. So that means myosin now cannot bind to actin. So that is basically what the troponin is doing during the resting phase, and because of this, the complete overlap between actin and myosin is not possible during the resting phase. I hope you understood the concept. Okay, so this was the actin, and now let us look at the structure of the next contractile protein, that is myosin. Now myosin again is a polymerized protein, that is, this is also made up of several smaller units called monomers. Now here we will see what are the monomers in case of myosin. So composition, it is made up of monomers called meromyosin. So several units of meromyosin combine together to form myosin. So how does a meromyosin look like? The picture which you see here on the screen, that is one meromyosin. So this is how one meromyosin looks like. So several meromyosins like this join together to form myosin. So now let us try to understand the structure of each meromyosin. Now each meromyosin structure can be divided into two parts, HMM, that is heavy meromyosin, and LMM, that is light meromyosin. So these are the two parts of the meromyosin structure. So heavy meromyosin consists of the head and the short arm. So this one is called short arm and this one is called the head. So this is the head, and this you can consider as the short arm. So this head and short arm are together known as HMM, that is heavy meromyosin, and this portion is the tail, which is known as the light meromyosin, that is LMM. So this portion is HMM and this portion is LMM. So here if you look at the structure, you can see that the HMM, that is the head and the short arm, are kind of projected outwards from the surface of the filament. So if you see, this is the surface of the filament and this head and short arm are kind of projected outwards. So this portion is also known as cross arm. Why is it called cross arm? Because it is exceptionally projecting out, so it is called cross arm. Now, this head of the meromyosin is very, very important, and it plays the most important role in the process of muscle contraction. So, if you see, head has active binding sites for ATP as well as actin, because if myosin wants to bind with actin, the way actin has active binding sites, similarly myosin should also have active binding sites, right? So, what are the actin binding sites? Okay, so these structures which you see here on the head, these are the actin binding sites. So that means these sites can bind with the active binding sites present on the actin. And what are these? These are the ATP binding sites. Now, why do they have ATP binding sites? Because during the process of muscle contraction, we see that ATP plays a very important role. So, for that ATP is required, and so the myosin structure actually combines with or unites with ATP. So, this is about the structure of myosin. So, now if you look at again at those same puzzle blocks, suppose if this is your actin, so this is the active binding site for myosin. If this is your myosin, then this is the active binding site for actin. And now when these two will combine, they can actually join here. But that is possible only if troponin is removed from the actin binding site. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.